So it's a real pleasure to be here in this fantastic venue. I want you to imagine a future in which your crisp packet doesn't outlive your great-grandchildren. That's basically what we wanted to do in this project. Supplant was about producing a biocompostable material for replacing film packaging for food, and it's coming from plants. And what we were trying to do is come up with a material which has similar or even better properties than existing polyolefin and polyester films, but at the same time it doesn't have a disruptive effect on the current market systems or the natural environment. And I think you'll agree that those are a pretty tough set of criteria to satisfy. So what I'm going to do in this presentation is tell you how we've addressed those and also tell you what we're doing in the future. But before I go any further, I'd like to introduce the team. Obviously, I'm here representing the project, also leading the second work stream here, which is producing the films and characterizing their properties. The films are made from cellulose, which is coming from plant-based materials. And in the initial stages of the project, these were made by my colleague Paul Dupré and his postdoc Parveen. And they are experts on lignocellulose biosynthesis. So we were growing plants specially to make this, which I understand is not the way that we're going to commercially produce these. But as I'll explain in a few slides, these give us the tools that we needed to modify the properties of the cellulose to make it more suitable for our film properties. So work stream one was providing the input here. And then in combination with my postdoc Daniel, who left the project about halfway through to take a group leader position in Potsdam, Jing Wang joined here shortly afterwards, and my PhD student Patience Abugu. We've been making films looking at the water and oxygen barrier properties. And then very importantly, my colleague Jonathan Cullen and his ex-postdoc Samantha, who's now a lecturer in Net Zero in Manchester. They were looking at how we manufacture those films and characterize them and doing life cycle and economic analysis to make sure that the way that we're producing them indeed satisfies those criteria I was talking about earlier on. So that's how it fits together academically. We're also partnered with PepsiCo and Beko, who are consumers of these films in future. Obviously, PepsiCo interested in snack foods. Beko looking at white goods, so preserving those in transit and generally keeping them away from oxygen and water. And you can't quite see the logo here, but this is Cambridge and Peterborough Recycling Partnership, or RECAP, who are advising us on end-of-life things, similar to what Mark was talking about earlier on. And NIAB is the National Institute for Agricultural Botany, who are growing the plants that we're using for our feedstocks. So why packaging? I think everyone knows why we target packaging, but I think this figure from Jonathan and his co-workers summarizes it nicely. It's not quite fully showing on the screen here, but the packaging represents a very substantial chunk of where the plastics flowing through the UK economy end up. So it's about 2.2 megatons per annum. So that's a figure I'd like you to remember. And a substantial proportion of that is film-based, which is quite hard to recycle or reprocess. It's difficult to clean. It's difficult to separate from organic matter. And so our idea was to develop a biocompostable material to replace those so that you don't have to do that segregation and washing and put everything ideally into an anaerobic digester. As we went through the project, we thought more about this in terms of carbon, and Mark has already mentioned about where this carbon cycle goes from feedstocks all the way through to back into the environment. And it was highlighted actually as one of the sustainable development goals for COP21 in Glasgow. And with our materials, we thought about how that's going to affect the amount of carbon being released through the existing waste streams, with the packaging coming from fossil fuels, where a lot of that is sent for landfill and burning, whereas ours is hopefully going to go round in this big loop. So here's a summary of sort of things we were doing. As I said, we're starting out by reducing the plastic pollution. Because we're coming from natural feedstocks, essentially, if everything goes well, we should be carbon neutral. There's an important caveat on that, which is obviously we need to do a life cycle analysis. And what we've found is that actually the production of our plastic films from those cellulose plant-based feedstocks, kilo for kilo, it's about 4 times the carbon impact of producing PET from a fossil-based precursor. But that is mostly dependent on one step in our process, which I'll talk about when we get to that. So it's highly optimizable. And also, we're not paying a price here for converting the biomass into oil first before it's then converted back into plastic. So we're going directly from the biomass to the plastic material. So you have to bear in mind those two caveats. I've said here that the feedstocks eventually are going to be sustainable. They're coming from forest resources and food and agricultural waste. So we're not going to be growing plants or especially producing our feedstocks just to make the plastic films. These will be coming from existing waste streams. So the applications, obviously, I've mentioned contaminated food and closed-loop systems like business-to-business manufacturing. So here's the layout for the project. As I mentioned before, the production of the feedstocks is taking place here in biochemistry. We take those through to the fabrication processes here, and then we feed back to work stream 3. All of the processing and the production of the feedstocks will go into the life cycle and economic analysis in work stream 3. So a little bit about cellulose. I know many of you may know about this already, but for those of you who don't, cellulose in plants is quite a complicated material. It's not just the crystalline polysaccharide. You have this kind of amorphous glue-like stuff called lignin, which binds everything together. You also have these molecules called hemicelluloses, which bind to the cellulose crystals within the plants. So in order to go from a plant to a processable material, there's quite a lot of steps that you have to do. So what we do is take away the lignin, but what we don't do is destroy the hemicelluloses. So in the traditional processes like the viscose process, you would break down the cellulose, even the crystals here, you would disassemble them completely into individual molecules and reconstruct them. It's a very energy and intensive and environmentally damaging process. Ours is not like that. We are using only very mild chemical treatments and enzymatic treatments, plus a bit of genetic modification. So Paul and his colleagues can genetically modify some of the plants that we're growing in order to produce mutants that have a slightly different distribution of charges here. These stars represent the cellulose chains, and by altering the distribution of charge groups on the surface, you can actually change the interaction between the cellulose and the hemicellulose, and that makes these crystals much easier to process and gets around this issue of having to regenerate the cellulose, which is what people like Courtaulds were doing 100 years ago, but our process is much more energetically and environmentally efficient. Okay, so I'm going to be showing you results later looking at both the wild-type natural cellulose and also some genetically modified stuff that we've used to move this process forward. The plants that we've been growing have been grown for us by NAB, which is located just outside Cambridge. If you haven't been there, they have an innovation farm which we've exhibited our materials at, and we at one stage had fields of what I call the world's most expensive cabbages growing in these things here. These were genetically modified brassicas which produced the initial feedstocks that we did our lab tests on, and they've been really helpful in making the initial stages of our feedstocks for our plastics. So obviously in the long run, we can't be growing our feedstocks in fields like that. So Samantha has been doing a lot of research here into what the available types of biomass are here in the UK. And these are summarized here. They're coming mainly from primary and secondary forest residues, agricultural residues, and some industries that use agricultural products. And the bottom line is that there's a lot of stuff there. We estimate about 2.7 megatons of lignocellulosic-based waste can be moved sustainably from existing disposal routes to bioplastic production, and that's without disruption of the main other uses. So I understand that a lot of these wastes are already purposed, but this is the fraction that we estimate is in excess. And if you remember a few minutes ago, I showed you that the total usage in the UK for plastics packaging is about 2.2 megatons. So this is easily enough to supply all of the film packaging if we decided to do that. So this is not a show-stopping problem for our materials. Okay, so how do we actually go from the plants to the feedstock? So we take our cabbages or the brassicas that we've been growing, and then we put them into solution here, and then we have to take away the lignin, which is the glue inside there, and we use a chemical called peracetic acid to do that. So this is what they look like after delignification. This is the step that probably costs us the most in terms of the CO2 impact. The production and usage of the peracetic acid is quite difficult. So that's the big step that is causing us issues from a life cycle point of view. Everything else here is in the noise in terms of its impact on the life cycle. And then we can either just take that as is without any treatments. We can do a chemical oxidation here, which is known as TEMPO. That is a complicated acronym which you don't need to know, but it basically produces charges on the surface of the cellulose crystals by chemical oxidation. And then we can also use an enzymatic method, which does the same as the genetic modification, but it selectively cuts some of the groups on the surface. And this is what we would be using to treat our large-scale agricultural waste, because obviously we can't genetically engineer those. And then there's a mechanical defibrillation treatment before we end up with a solution here of cellulose nanofibrils, and this is what it looks like, a kind of gloopy, like PVA wood glue or something, but more like with the viscosity of water. And then we can cast that onto a film here, and if you look in the TEM, you can see the individual cellulose nanofibers on the surface here. The scale bar is about 200 nanometers. So this is showing the process, how it works for the alpine flowering plant and Arabidopsis. This is the one that we used first. It's low volume, but we can control the genetic sequence there very well. Once we've understood it in Arabidopsis, we took that through to the brassicas and scaled up the amount of material. And now we're able to do this on kilogram quantities of waste from agricultural or forestry products. The defibrillation process, we're using here some grinding. So this is a bit like a kind of coffee grinder, and you grind up the material, get rid of all of the cells and other things. Usually about 40 to 50% of your plant is the useful fraction of cellulose, and then you need to dissociate those cellulose fibrils. So this can be done actually with a kitchen blender. We use the microfibrillator here, but you can do a similar thing. I actually did a demo in schools where we made some of these films just with a coffee grinder and a kitchen blender. It worked pretty well, actually better than I was expecting. But the point is to show you here that these fibrils here are highly dissociated, and that's the key to the success. In a minute, we're going to pass around some materials, and you'll see that they're quite uniform and transparent. And the main way that we do that is by ensuring a very, very good degree of fibrillation. And that's only really possible because of those treatments that I was talking about earlier on. Okay, so that's a little bit more about the processes that we do, but in view of the lost time, I'll skip that and go on just to show you the solution. So this is a solution here of the as normal cellulose. This is about 1% by volume distributed in water. And then this is the GUX mutant with the altered surface charge density. And there's not much difference really in terms of the opacity and how they look like visually. But you do notice a difference when you put them into films, and the way that we're making the films is to pour that into a Petri dish and then evaporate. This is obviously easy to do, but it's quite slow. We can speed that up a bit by using vacuum filtration here just to suck away a lot of the solvent, because obviously with such a large volume of solvent, we have to get rid of that quickly. So neither of these two methods are industrially scalable, but my last slide will have something more to say about how we would do this actually in industry. Okay, so moving on then to the properties, and at this stage, if I could ask Nisha, you could pass around the film. So this is a film that we've made from our cellulose nanofibrils, and in order to make it foldable, twistable and rollable, we've added some plasticizer here, which is hydroxypropyl cellulose, or HPC, about 40% by volume, and this just gives us that extra bit of plasticity that we might need for a film. And it's pretty transparent, not as transparent as PET, but it's still good enough, I think, for most consumer applications. It has good modulus and strength, actually probably even better than PET, and it's definitely something that you can process into a film covering. However, more, or at least equally importantly, is to increase the hydrophobicity. So one of the problems with cellulose is it's very leaky for water, and of course we want to make sure that water doesn't get in or out of our samples. So sorry about all this busy stuff here, but this is a comparison between our materials here. This is the hollow cellulose nanofibers plus a bit of ethyl cellulose. This is the raw cellulose nanofibrils, and we're measuring the water barrier properties using this machine here, which is used in the fiber industry to put a coating on, and we're putting a small coating, about 20 microns of this ethyl cellulose on the top to give a bit of extra hydrophobicity. And then by measuring both the oxygen and the water transfer rates, we can see here how the ethyl cellulose improves the properties of our material. So the WVTR here is the water transfer rate. The lower the better. This is the oxygen transfer rate. So you can see that cellulose is already pretty good. For oxygen transport rates, it's even better than PET, measured under different conditions. So under 90% relative humidity, this is a very stringent condition here. It's still better than PET. Normally, it's not so good if you compare it with PET on the water. PET would be down here about 10. Okay, so the as received holocellulose is much worse than PET. But with the ethyl cellulose coating, we can get it down. Some of our best values here now are about 23 grams per square meter per day. This is quite a difficult unit to visualize, but if you take a wine bottle full of water and spread it over a football pitch, that's about equivalent to 1 gram per square meter per day of evaporation. So 23 wine bottles poured over a football field is not a huge amount of water to be transmitted. And that is probably good enough for products like nuts or cheese and other kind of dairy products. And I think, Nisha, you can pass around the other demo here. So this is a sample that we've prepared here. Please don't eat the nuts. I think there'll be alternative refreshments at the coffee break. So these are just examples of some hygroscopic stuff that we've made. And you'll also see in there a little yellow disc. This is our material that we've dosed with a bit of curcumin, which is a pH-sensitive dye material. So this is just to give you a bit of smart functionality. In particular, if you have things like seafood products that degrade here, they change the pH, and this will shift the color here as they degrade. So you could have, as well as the barrier properties, you could have a sensor inside there which is telling you when your food is going off there. So that brings in a bit of the smart functionality as well as the barrier properties. Okay, so the last little bit here is about the large-scale manufacture of the films. As I mentioned before, these filtration methods, they're not good for scaling up. We're using too much solvent, they're batch processes, they're not good at scaling size, and they take a long time. So we have actually made some big steps to address that. Unfortunately, because we have a patent application pending, I can't tell you all about that, and I hate it when people say that, but it really is true. But I can tell you that we can produce, using this method, uniform films of arbitrary area, and they grow at about 1 micron thickness per minute, which, if you're trying to produce a 10, 20 micron film, is quite quick, much quicker than it takes with the solvent casting and vacuum filtration. And then the other nice thing is that they only have about 10% of the solvent remaining in the film. So this means that you can just bake that off quite rapidly. So these things are, I think, industrially scalable. And the barrier and the mechanical properties appear the same as the solvent cast film. So we're really happy about that bit. What I can tell you is we tried lots of other things which didn't work. So these are all the things that don't work. Roll-to-roll printing, unfortunately our solutions are not viscous enough to allow us to do that. Slurry extrusion, there's just too much solvent, and again, you get a collapse of the structure. And also direct extrusion would have been nice, but cellulose is a very heat-sensitive material, so that can't be done. So lots of traditional processing routes don't work, but we're quite pleased with this one as a viable alternative means. Okay, so let me just sum up there and put up the key points of what I think we've done. So largely, I think we've met the goals that we set out to do. We are not ready to go to full commercialization yet. Our materials are still too expensive, and the LCA is not good enough compared to pure PET. But I think we have got prototype materials that meet the mechanical and barrier criteria for food packaging. We've got smart environmental sensing, and I think we've shown, I hope, convincingly, that there's going to be low economic disruption and compatibility with existing agriculture and forestry processes. So we're not going to be creating any perverse incentives. And there is the scale here, if we wanted to replace large volumes of film materials. So I hope you found that interesting. If you have an idea about how we can develop this further, very interested in looking for funding or further collaborations. And thank you again to all our partners, and especially to the SSPP team, to Paul, Nick, Ashley, Heather, and others. You haven't just funded us, you've supported us, you've nurtured us, and I've really enjoyed working with the SSPP team. So thanks a lot.